So between array normalizations are important if we have more arrays within one experiment, or maybe we are comparing different experiments or different treatments within the same experiments with one another. We compare samples in different arrays. Hybridization reactions may vary. We need to correct for variability among the arrays, or variations are due to some experiments. So first type is we call it as centering normalization. So the means of all distributions are equal. So we assume that all distributions, they have equal means. So what we do is we subtract the array mean in a log ratio or intensity from each feature. So we take the mean for the array, we take the mean for each feature within that array, and we correct every mean for every feature. We normalize it for the overall array means, and then across different arrays, since we are assuming that means are equal, so when we do this normalization, we believe that across different arrays our data is normalized. So all arrays will have then an equal mean or mean of 0 after this normalization. Alternative is sometimes we probably want to use median because means are prone to outliers. So if we have outliers in our data set, they will pull the mean towards them. So very big numbers, the mean will be skewed towards the big numbers, and if there are more small numbers in the data set, since mean is the average, so that is skewed towards those small numbers, whereas the median is the middlemost value. So we can use median rather than mean if we have the issues of these skewness. So in scaling normalization, the means and the standard deviations of all distributions are equal. So we assume equal variance, equal mean assumptions, and we also call this process as standardization. So what we do is we subtract the mean of array from the features and divide it by standard deviation. So in this way we are coming up with some numbers with some values. We call them as z values or standard values, or we have taken this thing earlier before in z distributions. We call it as a standardization procedure also. So the mean of the measurements on each array then will become 0. The standard deviation will be 1 after this scaling. As I said earlier, we can use median and median absolute deviations, MADs, rather than means and standard deviations. But mean is preferred, since for computing distances and correlation coefficients means are used, so that's why we probably want to stick with the mean. Quantile distribution is we take the distributions of the data on each of the array and we assume they're identical. So in quantile or distribution normalization, we standardize the data, and for each array we ordered centered measurements from lowest to highest. Then we compute a new distribution. We use the average across all arrays for each ordered feature, and then we replace all measurements with the corresponding new average. For example, we have 1, 6 and 7 for G1, which is actually our gene 1. A1 is for example our treatment 1, or within treatment A we have replicate 1 and then A2 and then A3. For gene 2 or feature 2, we might have 2, 5 and 8, and for 3 we have 3, 4 and 9. We rank them. So for each gene or each feature, we rank them as from lower to higher. We order them 1, 2, 3 goes in A1, 4, 5, 6 in there is an order you see guys here, and then we have 7, 8 and 9. When we take the averages for each rows, for the top row the average is 4 and then 5 and then 6. We use these averages and we correct every number for these averages, and finally normalized numbers, they are like this. So if we do this quantile distribution normalization, quantile or distribution normalization, what are the effects on the data? So here we see, for example, you have different distributions which may be the replicates within different treatments. So green is one treatment with 5 replicates, or these are different arrays within the same experiments, you can also say that. Then we have yellows, red, blue and so on. So in this way, if you look into all these sets, we are looking into that for, though the treatments are the same, but for each of them we see those, we have big variances. The boxes, their medians are quite far off from each other. You see those reds, you see those blues. and so on. So here we see after normalization, the distributions belonging to particular treatments. Here we have those 5 different colors. So all those distributions belonging to each color, that may be, as I said earlier, the arrays belonging to one experiment and the other, or they might be within the same experiment you have arrays which are representing different replicates. So we see these distributions are now pretty close to one another. If you look at the top, those dark circles, they are your outliers, the extremely high numbers. So to compare samples in different arrays, we do these between array normalizations. These hybridizations reactions, they may vary. We correct for variability among arrays, and sometimes those variations may be present because of these different experiments.